Hello everyone, welcome back to Dr. K. Prem-PRIMER lecture series presenting by Dr. K. Prem, that's me. In today's episode, we will talk about beta galactosidase or beta galactosidase of E. coli. It's also called as lactase. The lactase or beta galactosidase break down the disaccharide lactose into galactose and glucose molecule. In today's lecture, we will focus on what is the operon structure and especially beta galactosidase gene structure, and followed by biochemical function of beta galactosidase as well as the protein structure of beta galactosidase. And finally, how this beta galactosidase was used in the construction of various cloning vectors for screening of recombinant clones, right? In the here, first we will see the operon. So operon is a known concept in the prokaryotes, where metabolically related genes are clustered in a region, and those genes are uses the common regulatory elements for regulations. Means they share the common promoters for upregulation or downregulation. They use the common regulatory elements. So the operon is the first such kind of operon was discovered and studied in E. coli. You can see in this one, this is a operon where the PI indicates the promoter of repressor gene, to which downstream there is a repressor gene, and this repressor gene product, it's a repressor protein, binds to the operator region of the beta galactosidase and permease and trans-acetyl transferase, and it inhibits the transcription of these genes in the presence of glucose. Whenever there is a lactose and in the absence of glucose, then this operator is operator, which will be from that repressor protein will be removed. Then there is a transcription. You see, repressor, and this is the promoter of operon. This is a PL, and this is a operator region where a repressor gene binds in the presence of glucose. When it is when the E. coli grows in the presence of lactose and absence of glucose, then repressor is removed from this operator and allows the transcription of LacZ, which encodes the beta galactosidase. That is nothing but lactase, which is involved in the breakdown of lactose into galactose and glucose molecule. Then the LacY encodes the lactose permease. This enzyme, this transporters protein goes and decorated in the plasma membrane and allows the transport of lactose from medium to the E. coli cell. And the next one is LacA, which encodes the galactoside acetyl transferase, whose substrate is still obscure. And today we are going to talk about and focus on LacZ, not on the lactose permease or other enzyme, right? You see, the lactose is breakdown by beta galactosidase, and whose gene is LacZ is having the 3075 base pairs, includes the ATG and stop codon. So length of the beta galactosidase gene is 3075 base pairs, and it encodes a protein which is having the two domains. Monomer have two domains, alpha domain and omega domain. Alpha domain is extended from first amino acid to 140 amino acids, and then the rest of the domain is called as omega domain. So the total amino acids present in the each monomer of beta galactosidase is 1023 amino acid, and its molecular weight is 116 kDa. So the beta galactosidase functionally active whenever it forms the tetramer. Whenever four molecules of beta galactosidase associates and form a tetramer, that's a functionally active. And you see the beta galactosidase having the tetramer, and then the molecular weight is 464 kilodaltons. And the tetramer requires the both magnesium and calcium, magnesium and sodium cations for its activity. As I told you that tetrameric beta galactosidase is a functionally active, which acts on lactose, which is a disaccharide, right? So disaccharide means here you see the lactose is having the galactose and glucose, and these two moieties are connected by a glycosidic bond. That's a beta 1,4 glycosidic bond. That lactose is having the beta 1,4 glycosidic bond. So the beta galactosidase enzyme acts on the beta 1,4 glycosidic bond, and converts into two moieties. One is the galactose and the glucose. At the same time, beta galactosidase also have a one more property that is called as isomerization. Means it will convert the lactose to the yellow lactose, which is sharing the beta 1,6 glycosidic bond. You see the bond is 1,4. When they have a 1,4, that's called as lactose. When the bond is beta 1 to 6, then that is a yellow lactose. Whenever you supply lactose to the E. coli, then what happens? The lactose is some extent of lactose is converted to the yellow lactose. This yellow lactose binds to the repressor gene, repressor protein and it will become inactive. Repressor can bind to the operator as well as the yellow lactose. So whenever yellow lactose is formed by the beta galactosidase, then this yellow lactose binds to the repressor and it will inactivate the repressor. At the same time, yellow lactose bound repressor can dislodge from the operator region, and once the operator region is free, the RNA polymerase binds to the promoter and transcribes the LacZ, LacY, and LacA. So this is what is the significance of yellow lactose. Yellow lactose is a natural inhibitor of repressor molecule. This is about the biochemical function of beta galactosidase. And beta galactosidase also have many substrates. Like one of them is X-gol. That's nothing but 5-bromo, 4-chloro, 3-indolyl beta galactopyranoside. This is the compound. So on which it will act, and it is having the galactose moiety and indole moiety. So whenever beta galactase found act on the 5-bromo, 4-chloro, 3-indolyl galactopyranoside, and it break down into the two moieties, that is galactose and indole. Galactose and indole. So whenever indole is released, the indole is immediately dimerized and produces the blue color precipitate. So whenever you grow E. coli, which E. coli in a lactose media, and if you supply the X-gol, so the colonies looks like blue in color because it can break down the 5-bromo 4-chloro 3-indolyl galactopyrenoside into galactose and indole molecule. The free indole molecule is dimerized and forms a blue color compound. Blue color compound. So this hexagon is used in the selection of recombinants. Right. So now we'll move to the next session where how beta galactosidase was used in the construction of cloning vectors and how it is useful in the single step visual selection of recombinants. As I told you that one of the important property of the beta galactosidase was critical in the development of pUC18 vectors or pUC series of vectors. So that is, alpha, as I told you that beta galactosidase have two domains, that is alpha domain and omega domain. So if you fragment the beta galactosidase into alpha domain and beta domain, omega domain. So whenever you fragment beta galactosidase into alpha domain and omega domain, so what will happen? These domains independently, they don't have the beta galactosidase activity. But if you can mix the both alpha fragment and omega fragment again, and they can form a tetrameric beta galactosidase, and this restores the beta galactosidase activity. So if you fragment the beta galactosidase into alpha and omega, they don't have a independently, they don't have a beta galactosidase activity. But if you mix them, both the fragments, it can form a tetrameric beta galactosidase and restores the beta galactosidase activity. This property they observed with the beta galactosidase, and based on this property only, pUC18 vectors are designed to screen the recombinant clones. I will show you how. Here, as I told you that if you associate both of them, and it restores the beta galactosidase activity. As I told you that the alpha region is encoded by the LacZ region, LacZ prime region, and the omega region is encoded by the LacZ, mutated LacZ. So if you can incorporate the alpha peptide encoding LacZ prime region into the pUC vector, right? pUC vector, you see, this is a pUC18 vector. The LacZ region encoding DNA fragment is incorporated the pUC18 under the downstream to the PLAC promoter, right? So that LacZ prime encodes the alpha peptide whose length is almost 30 amino acid. You see, so the 30 amino acid, as I told you in the beginning, the alpha peptide is extended up to 140 amino acid, but here we are using only 30 amino acid encoding region only, that is incorporated the pUC18 vector. So alpha portion that is encoded by the LacZ prime encodes a N-terminus alpha peptide, that's a length of 30 amino acid of beta galactosidase, is incorporated to the pUC18 vector. In that, there is a multiple cloning site, allows the cloning of various fragments by using the restriction enzymes, and it is also have ampicillin resistance marker and origin of replication. So the main point I want to highlight is that the alpha region, alpha fragment encoding LacZ prime is incorporated to the pUC18. So one portion of enzyme is in the pUC18. Then other portion of the enzyme, that is omega encoding LacZ region is there in the E. coli genome. E. coli genome. So one part, N-terminal alpha peptide encoding region is present in the pUC18 vector, and the remaining LacZ region, mutated LacZ omega region, is a encoding region present in the E. coli genome, right? So if you, whenever you transform this pUC18 carrying the LacZ region into the LacZ delta M15 E. coli string, then that bacteria can form a functional beta galactosidase. And how do you know the functional beta galactosidase form? If you grow that bacteria in the presence of X-gal, it forms the blue color colonies. So alpha region is present in the vector, and omega region is present in the E. coli host. So whenever you transform this pUC18, then these E. coli can form a blue colonies in the presence of X-gal. So this is what is the basis for the alpha complementation, right? So now you see this is the LacZ delta M15 E. coli strain, which is having the only omega region encoding region, which encodes the omega peptide, right? So this bacteria, if you transform with the pUC18 vector, right, then what will happen? So the pUC18 vector have a LacZ prime region, and which encodes the alpha peptide. So the alpha peptide from the pUC18 and omega peptide from the genome, and both are associated and form a functional tetrameric beta galactosidase. So such, whenever the bacteria with the bacteria which is transformed with the pUC18, if you grow these E. coli on a media with X-gal and IPTG, such colonies looks blue in color, right? So because the functional tetrameric beta galactosidase is formed with the help of genome as well as the vector. But this is all because the vector is self-ligated or undigested vector. If you clone your gene of interest into the LacZ region through MCCS, multiple restriction site, what will happen? We'll see. So whenever you E. coli strain is transformed with the vector plus insert construct, construct. So what will happen? We'll see. So usually E. coli will produce the omega peptide, and here this construct is, you see, this construct is vector plus insert. Insert is inserted into the LacZ prime region through the MCCS restriction enzymes. So when LacZ prime region is disturbed with the insertion of your gene of interest or a DNA fragment, then what will happen? The alpha peptide, which may not be produced from this one, or it may be produced with the different configuration or conformation. So if it is not producing, then there is no association. If it is alpha peptide is produced with a different configuration, this alpha peptide, which is lost its configuration, cannot bind with the omega peptide. You see? Then there is no functional tetrameric beta galactosidase when E. coli strain is transformed with the vector plus insert. So these kind of E. coli cannot metabolize the X-gal. So the colonies looks white in color when you grow them on a media with the IPTZ and X-gal. So here you see the blue colonies are non-recombinants, and white colonies are recombinants. So the visual selection of recombinants and non-recombinants is all happens with the help of beta galactosidase. That's what is the contribution of beta galactosidase for the development of cloning vector, that is a pUC18 and other. Hope you understand the concept. And I'm going to ask you one question on this same concept. Which one of the following is a natural inhibitor of repressor protein of the operon? So, which one of the following is a natural inhibitor of repressor protein of the operon? IPTG, yellow lactose, and lactose or XGOL? Please answer this question through the comment section. If you have any question, let me know through the comment section. I'll get back to you once I see your comment. So thank you all for listening the beta galactosidase. And if you have any other questions also, please let me know. I'll get back to you. And try to subscribe for regular updates. And if you like this concept and the presentation and contents, you can share with your friends, no issues. And don't forget to give a thumbs up if you watch it and like it. And also write to me your doubts through the comment section. I'll get back to you. So don't forget to subscribe my channel. That's Dr. K PREM-PRIMER. This all about, I'm going to talk about only RDNA technology soon or later. And maybe extend it to other subjects. But right now I'm talking on only RDNA technology. Thank you for watching. See you soon with the other concept. Until then, bye-bye.